Today I would like to talk about the cancer-associated sugar chain modification and using this evolutionary strategy to target the cancer cell in childhood malignancy. Sorry. So as an introduction, Anopheles mosquito bites the human host where malaria parasite infect the erythrocytes, and normally these infected erythrocytes are cleared through the spleen. But when pregnancy-associated malaria happens, these infected erythrocytes are going through the placenta, where these parasites present the Htoo CSA protein on the surface of the infected erythrocytes to attach at the placenta by binding to a distant type of chondroitin sulfate A. So before going to experimental result, I would like to reintroduce the phenotypic similarity between the placenta and the malignancy, because they are having the re-expression of the less differentiated stage, invading into the surrounding structure and rapid proliferation rate. With our finding, we conclude that these placenta and malignant compartments are sharing a similar Oncofetal CS signature. So we validated that these infected erythrocytes can bind to the human cancer cell, and they can be inhibited by purified CSA. And also, it is validated that these Duffy-like binding region flanked with the interdomain is a minimal binding region which has a similar binding capacity to the wild-type malaria protein Htoo CSA. So we are using this recombinant Htoo protein to detect the cancer cell binding, and we have seen strong binding with the lymphoma, osteosarcoma, and rhabdomyosarcoma, and these are dose-dependent in nature. And here, vivo cell line, which is a choriocarcinoma cell line, which is widely used for as a model for the villus trophoblast function, and we can see the binding can be inhibited by putting purifying CSA. So these Htoo can bind the human cancer cell line, but it doesn't bind or it only weakly bind to the normal counterparts. So I would like to explain about what is the difference between the Oncofetal CS and normal CSA. So normal CSA, we can be exposed by treating with the enzymatic digestion to expose the 4-O-sulfation group, which can be detected by the monoclonal antibody. Here you can see C4S monoclonal antibody. So when we compare these two binding, like such as in placenta, we can see the binding with the both antibodies, but normal CSA is expressed throughout the normal tissues, but Htoo can only exclusively present in the placenta of the normal tissue. So here's the system we use for the detection. One cancer cell or placenta has a CSA that a malaria Htoo protein can bind, then we can be detected by conjugated V5 and secondary antibody reaction. So now we know that it can bind to the cancer cell. So we screen the normal counterparts in the pediatric normal tissue, and we have seen very few or absent binding in the normal tissue. And here again, so we can inhibit the binding of the Htoo by putting the purifying CSA or chondroitinase treatment. We also screen the adult human tissue, and Htoo showing the strong binding only in the placenta. Here you can see the trophoblastic binding of the VAR2 in the human placenta, but not in the other normal tissue. So as a standard cancer St. Patrick Dream Team project, we have screened a lot of sarcoma and brain cancer tissue. And here you can easily see normal organ TME has a lot of negative codes that can be seen by blue area. It is only nuclear staining. And where other sarcoma tissue array have a lot of positive staining result. So this expression is, we counted for the % positive core across the TME. Expression can be seen as in the cancer cell or cancer-associated stromal compartment. And so we score as a cancer cell compartment or cancer-associated stromal compartment. So because these sugar chain modification is not only in the cancer cell, they can be in the extracellular matrix. So extracellular matrix is known for the signaling events that can be associated with the tumor proliferation and aggression. Now I would like to emphasize on the neuroblastoma TME, which has tissue microarray, which has a good follow-up clinical annotation data, so we can analyze the Oncofetal CS expression and tumor characteristics. And we found that CS expression is significantly associated with the advanced tumor stage, and poor survival prognosis. Now I would like to talk about the collaboration projects with the John Maris group. And I think now everybody know about what is glypican-2 as introduced by the Krista Michael earlier. So it has been known that glypican-2 is associated with the heparin sulfate binding. So our question here is, this glypican-2 is also Oncofetal CS modification present or not? So to address that question, we use proximity ligation assay, which is a simple technique. Just we put the 2 primary antibodies, and then PLA probes that has a synthetic DNA template, and it can start a rolling thermocycle reaction at ambient temperature where these 2 epitopes are close enough to each other in proximity. So here we can see glypican-2 incubated with VAR2 primary antibodies. We can see the signal in red color, and where we can see the signal with the DBL4 or control group or glypican-2 alone. So from this result, we can conclude that glypican-2 is likely to be CS modified, and our next step is to emphasize, this is the C32 melanoma cell line, because we found that protein on VAR2 pull-down assay on melanoma cell lines. And now glypican-2 has a very good RNA data on neuroblastoma. So our group is eager to forward with the neuroblastoma cell lines and tissue microarrays to address that question. Now we know that VAR2 can bind the cancer cell, and our next question is whether it can be internalized into the cell or not. So here we can see green color is VAR2 with immunofluorescent image, and you can see here it can internalize into the cells. And this process can also seen in other different cell lines. And so our next question is how that VAR2 can be uptake into the cell nucleus or in the cytoplasmic. So here we conjugated VAR2 with a gold particle, and using the electron microscopy, we see that there is possible caveolin uptake, but when we use the caveolin inhibitor, but it still goes into the cell. So it is not only the one mechanism-dependent internalization. And when we conjugated these VAR2 with heme-acetylene, which is a tubulin inhibitor that can inhibit the cell cycle, and we can effectively kill the sarcoma cell lines with the low picomolar or nanomolar concentration. And we also tested that tubulin inhibitor function still preserved in the VAR2 conjugated drugs or not. And this drug is now co-developed by VAR2 Pharmaceuticals and Kairos, and it is now in preclinical developmental stage. We also screen more than 30 cell lines, and it shows it can effectively kill the cancer cell with a very low 2 picomolar concentration to 30 nanomolar concentration. So our next question is, it still can effectively kill in the mouse model xenograft or not? And we addressed that question with the lymphoma and prostate cancer cell line. And here you can see VDC compared with the toxin alone or vehicle is effectively kill the cancer cell. And there is no adverse side effects on the kidney or liver. So far, these drugs are tested on the human cell, xenografted into the mice. So it might not affect the mouse normal organ. So for this, we developed the 41, which is a mouse memory tumor cell lines, and we injected into the left ventricle of the mouse for the bone metastasis, and overall penetrance is 50 to 60%. And here we stain with VAR2. Sorry, here we stain again with VAR2 for the Oncofetal CS expression, and we can still see in the bone metastasis compartment. And at the end of the study, VDC-treated group mouse are still alive and no metastasis, while other control group, toxin alone, die with the metastatic disease. So our future steps is to doing the rhabdomyo and osteosarcoma orthotopic model, and now we are getting the ethic approval. With that, I would like to thank everyone in my lab, and also Stand Up to Cancer Foundation for the collaboration and providing grant. And thank you, everyone.